Hello friends, in this module we will be talking about fluorescence microscopy. We will cover components and their functions, the working principle of fluorescence microscopy, ray diagram, applications of fluorescence microscopy, advantages of fluorescence microscopy and limitations of fluorescence microscopy. To understand how fluorescence microscopy works and why it has become so important to modern biology, one must first understand what the term fluorescence means. George Stokes observed that mineral fluorspar emitted red light when illuminated by UV light. This property of certain substances to emit rays of a different wavelength within nanoseconds after absorption of light is called fluorescence. Wavelength of light emitted is longer than that of excitation light, and the difference between the exciting and emitted wavelengths is known as the Stokes shift. By completely filtering out the exciting light without blocking the emitted fluorescence, it is possible to see only the objects that are fluorescent. In this picture, you can see bacteria glowing against a dark background. Let us first discuss the principle of fluorescence. The atom is initially in the ground state. Energy is absorbed by the atom, which becomes excited. The electron jumps to a higher energy level. Soon, the electron drops back to the ground state, emitting light. We now say the atom is fluorescing. In fluorescence microscopy, the sample is itself the light source and has to be fluorescent. The sample can be either naturally fluorescing like chlorophyll and some minerals like fluorspar, or treated with fluorescing chemicals also called fluorochromes. Some of the fluorochromes commonly used are acridine orange, acridine yellow, auramine O, fluorescein, primulin, rivanol, thioflavin and rhodamine. In fluorescence microscopy, the preparation itself becomes self-luminous, whereas the radiation used for excitation does not contribute to the image formation but is actually eliminated by barrier filters. For excitation, the radiation of shorter wavelength than that emitted by the sample preparation is used. Thus, fluorescein, which emits green fluorescence, can be excited by blue light, and rhodamine, which emits red fluorescence, by blue, green and yellow light. Let us now see a few fluorescent microscopy images to get you excited. Green fluorescence, red fluorescence and yellow fluorescence. Let us now talk about the instrumentation of the fluorescence microscope. Apart from the basic components which we have studied for other microscopes, fluorescent microscopes require several light filtering components. These specific filters are needed to isolate the excitation and emission wavelengths of a fluorochrome. The first filter is excitation filters. These are also called primary filters and are placed between illumination source and the objective, as you can see in the figure. The exciter filter passes only the desired excitation wavelengths, like in this case only violet or blue light is allowed. These rays are reflected down through the objective to illuminate the specimen. The second filter is barrier or emission filters. These are also called secondary filters and are placed between eye and object. Allows only emitted light to pass and filters other wavelengths from reaching the eye. Like in this case, only red wavelength is allowed and violet is blocked. As a result, only a single colored image is seen at a time. Let us now talk about another filter or component of fluorescence microscope, the dichroic mirror. The dichroic mirror is also called chromatic beam splitter. It reflects light of some colors but transmits light of other colors. In this figure here, blue light is reflected whereas red light is transmitted. It isolates the emitted light from the excitation wavelength. The dichroic mirror is to be selected in such a way that wavelength at which a beam splitter allows the longer wavelengths to pass must be set between the excitation and emission wavelengths of any given fluorochrome so that excitation light is reflected and emission light is allowed to pass through it. Let us quickly revise the three important filters of fluorescence microscope. The incoming light waves are filtered by excitation filters. This is the dichroic mirror which allows light rays of only selected wavelength to pass through it. This is the emission or barrier filter which allows only the emitted light or fluorescence to pass through it. Let us now discuss the ray diagram and working of the fluorescence microscope. As you can see in the figure, a high intensity source of light is used because only a small part of the incident energy is converted into fluorescent light. It might be a mercury or xenon arc lamp. It emits white light which falls on the exciter filter. The exciter filter is such that only the blue light is transmitted through it, whereas all the other colors are blocked off. The blue light falls on a dichroic mirror which is placed at an angle of 45 degrees between the objective lens and the eyepiece. The mirror reflects only the blue light downwards to the specimen. The stained portion of the specimen treated with a fluorescent dye absorbs blue light and emits green light. This green light passes upwards and is transmitted through the dichroic mirror and finally reaches the barrier filter. This filter allows only the green light to pass to the eye and blocks out any residual blue light. Thus, the stained portions of the specimen appear as glowing green against a black background. Let us now talk about the applications of fluorescence microscopy. Fluorescence microscopy is a rapidly expanding technique both in the medical and biological sciences. The technique has made it possible to identify cells and cellular components with a high degree of specificity. For example, certain disease conditions can be studied with the fluorescence microscopy. Some fluorescent dyes are specific for certain cells. For example, the dye fluorescein isothiocyanate attaches to cells of Bacillus anthracis and appears green when viewed under a fluorescence microscope. Another dye, auramine O, is specific for Mycobacterium tuberculosis and appears yellow. Thus, these organisms can be identified from any sample containing them. Fluorescent dyes bind to specific molecules like DNA. The picture here shows fibroblast cell line stained with host stain. Green fluorescent protein, that is GFP, from jellyfish can be fused to any gene, giving rise to recombinant protein that fluoresces green. Thus, live cells can be imaged. The picture here shows bacterial colonies transformed with GFP, and hence you can see the colonies glowing green. Fluorescence microscopy can be used to visualize and localize proteins or DNA within a cell. The picture shows mammalian cells double labeled. DNA can be seen as blue colored and microfilaments can be seen as green. It is also widely used in textile industry to analyze fiber dimensions. Fluorescence microscopy staining also is useful in the field of mineralogical applications. It is routinely used for the study of minerals such as coal, graphene, and others. Another application is in the field of serology, particularly immunofluorescence. When bacteria are injected into a host animal, the body responds by producing antibodies. The injected bacteria here acts as an antigen. The antibodies are coupled to a fluorescent dye such as fluorescein. When fluorescein labeled antibodies combine with bacteria and are viewed under fluorescent microscope, the organisms appear luminescent. This technique can be used to detect the causative organisms from the samples. Fluorescence microscopy uses are widely used in the field of histochemistry to detect particles such as neurotransmitter amines which cannot be seen by conventional microscopes. Let us now talk about the advantages of fluorescence microscopy. The most important advantage of fluorescence microscopy is its extreme sensitivity. It can detect as few as 50 fluorescein molecules per cubic micrometer. Techniques of fluorescence microscopy can be applied to organic material, dead organisms or tissue, or to living material with the use of in vitro or in vivo fluorochromes. It can be used for the diagnosis of specific diseases like tuberculosis, malaria, and anthrax. The results are quick and reliable. It is a method which can be used to study the dynamic behavior exhibited in live cell imaging. Different molecules can be stained with different color fluorochromes, allowing multiple types of molecules to be tracked simultaneously. No method is without limitations. So let us discuss about the limitations of fluorescence microscopy. Excitation light may damage live tissues. Certain biological structures such as mitochondria, riboflavin, and collagen fluorose after absorbing light without the addition of fluorochromes. This is due to autofluorescence, which can be problematic in detection of fluorescence probes in tissues and cells. A major problem with immunofluorescence is photobleaching. It can be described as the photochemical destruction of a fluorochrome due to the generation of reactive oxygen species in the specimen as a byproduct of fluorescence excitation. In diagnostics, photobleaching can result in wrong results. Higher cost and technical complexity. And the last limitation is the lifespan of fluorochromes is very short.